Greetings and welcome to EC Connect and Logan Startup Hub. I'm Brad Apps. In June at the Logan Startup Hub, we hosted an event about life sciences, highlighting some great things being done in this arena in Queensland. For those who couldn't make the event, we've uploaded them and made them available on the Innovate Logan website and YouTube channel. Before we start, I'd like to give you a quick update on what we do here at EC Connect and Logan Startup Hub. So EC Connect is a software company specializing in fully automated and high quantity billing and payment systems, as well as client management, data management and system integration tools. We've been providing these systems to the telecommunication industry for over 14 years and also have a large suite of telco provisioning and management facilities as well. The EC Connect team consists of developers, software analysts and system administrators, and due to the numerous IT platforms which our software runs on, we have much experience with Linux, AWS and MySQL. We've also helped numerous startups with our platforms, expertise and custom development. In 2017, we opened part of our office as a co-work space, offering the unique opportunity for startups to work alongside an existing successful company, and here Logan Startup Hub was born. The goal of our initiative here is to increase our presence in the community, share our experiences with new and upcoming businesses, provide a location to facilitate collaboration and the sense of community, and support local employment and allow people to work close to home. We also host events to help small business, entrepreneurs and startups. We are supported by Advance Queensland and Logan City Council. We thank you for their support. Now enjoy these presentations and we'll catch up soon. Thank you.
My name is Davies. I'm the CEO of Vale Life Sciences. We've been operating for about 3 years. We're established in 2017. We're based in the Translational Research Institute in the TRI, which is behind the PA Hospital in Brisbane. We were set up to commercialise technology developed by myself during my time at Trinity College. And our products are already used by scientists across the world. So what do? We're really focused on cell and tissue culture, and cell and tissue culture has a very broad range of applicability. It can be used for everything from making vaccines to research. In the research area, there's a big problem. So cells are used widely by researchers in every field, in cancer, various diseases, models, etc. One of the problems is that the cells don't give us the readouts that we generally see in the body. It's very difficult to predict whether a drug is going to have any real effect or efficacy when we perform these experiments using cells. So there's been a huge push over the last 10 years to make these models more predictive. After a significant amount of soul searching and analysis, the industry has sort of boiled this down to 4 key areas that need to be addressed. These are relevance. Does the work that we perform using cells mimic that in the body? 2, reproducibility. Can you actually reproduce those results again and again without? This is a key area and has been causing huge problems in the industry. And it's resulted in a huge loss of money, around about $24 billion in 2016 in the US alone. Simplicity. Scientists have all sorts of backgrounds and skill levels. We need to, the technologies need to be simple enough that anyone can use them, and cost. Obviously, the economic cost of experiments and research is always a key factor. We've got a number of products. So the first one is Happy Cell ASM. This is a liquid media. It's actually a non-Newtonian liquid, which has all the characteristics of a fluid or culture media, the media we use, but has a polymer in it which gives it a gel-like property. Now, what this allows us to do is it allows us to grow cells in 3 dimensions. And what we have here is a 2D. This is the way cells are typically grown at the moment in 2 dimensions. You can see that it's flat on a substrate. And we found that actually growing cells in 3 dimensions makes the experiments more in vivo-like. It gives a better relevance to the experiments. So what we have is a technology that allows us to do this easily and reproducibly and cheaply. If we look down here, here's an example of a tumor spheroid, a cartoon of a tumor spheroid, and here are some tumors that we've grown in the lab that mimic the structures very well. This is an ongoing area of research. It's a very, very hot area, and everyone wants to move into this area, but barriers are typically technical and financial. Our second technology is a microplate technology. Microplates are used in every aspect of biochemical and biomedical research, from the very early stages up until the clinics and beyond. It's the standard platform that we use for our experimental work. We've developed effectively a better system for this technology. So one of the big problems is that microplates are notoriously poor for reproducibility. Experiments performed on the outside wells of a plate will differ from other locations in the plate, which means that inherently you have a system that offers poor reproducibility before you begin. What we've managed to do, and this is a very busy slide, but what we've managed to do is actually reduce that variation substantially. And typically we can halve the variation in a microplate, which offers huge advantages. It means you have to do fewer experiments, half the number of experiments to get the same result. And it's just gives you better confidence in your work. This technology also allows us to miniaturize, and we're able to miniaturize experiments down by 1,000 times, which leads to enormous cost savings in the industry. There are also added advantages. Often experimental material is so scarce, especially if we're using samples from humans, that we need a way of miniaturizing those experiments so we can get more per sample, and this allows this as well. So I'll get into the opportunity. The cell culture market at the moment is worth around about $16 billion. Now that covers a lot of areas. It's projected to rise to 29 billion by 2024. The growth applications are biomedical research, tissue regeneration, vaccine production, and antibody and cell therapies. These are all areas in which we operate. We have applications for our technologies in all these areas. One of the big problems in this, with this is that this is a very large market, and it's guarded very effectively by the large multinational companies operating in this space. One area that we are aware of that is being poorly served by the multinational companies, and so far they have failed in any meaningful way to make an entrance into this market, is the 3D and microplate market for research. This part of the market is a lot smaller. It's worth about 200 million, rising up to about $1 billion by 2024. But this would allow us to gain entrance to the larger market. This is an area that we can definitely get into, and we're now targeting. The demographic, well, we're looking for researchers, obviously laboratory technicians, specialists, people operating drug companies, etc. And we're targeting obviously the pharmaceutical, biotech, academic and governmental organizations. Because of limited resources, we're focusing our attention to 3 geographical areas, and that's Australia. Obviously, it's our home market, and it's a good place to start. The UK, where we have people in our organization already embedded, and the USA, which is the largest single market in this area in the world. The sales and marketing strategy, I'm not going to get hugely into this, but we already have one. We've been operating, unfortunately, on a shoestring budget, but developing websites and trying to generate local uptake of our products with some success, although we could have done a lot better. We currently have distributors in Australia, USA, Japan, Ireland, and Singapore. And really, what we need our money for is to increase this activity. We've got some great technologies; they work extremely well. They knock the competition into a top 10, and we need to get these technologies out So really, where we want to head now is to produce is to take on a sales and marketing manager for these territories who has got a broad background in this area. Until the COVID-19 outbreak, our strategy was to attend conferences, which is where you get the best return on your investment for marketing, and also make visits to individual researchers, key researchers at universities and various drug companies, key opinion leaders in the field, who, through my experience as an academic for 25 years, have already know many of these people or their groups and have ways into this. We have a very comprehensive patent portfolio, all granted, all paid for, all granted, all finished, including 2 trademarks. Finally, our team, well, there's me. So I'm the CEO, CSO. I have a PhD in cellular physiology and 25 years experience. I've worked at several very large UK universities, including Dundee, Glasgow. I then moved over to Ireland at Trinity College at Dublin and spent 3 years here in Queensland working for QUT. Meredith Clark, she's the Chief Operating Officer. She has 35 years of business experience and management, worked for a whole range of multinationals. The other director here is Pat White. Pat is based in Galway in Ireland. He runs Irish Micromouldings, which is a company which makes plastics for the medical device industry, and it makes very high-end devices, medical devices, mostly for the cardiovascular medical area. He manufactures a lot of our plasticware, and he's also been an investor for the last 3 years.
Excellent, Anthony. Very good. Thank you so much. We'll ask a few questions now, if that's okay.
Yeah, sure.
So it's all about really testing new molecules, is it, or testing new cells, or what is your technology about? Is it more on the molecules or cellular level, or both?
So what we're offering is so tissue culture and cell culture. It's growing the cell, but what we're offering are very new technologies in this area. So the way people grow cells and tissues, or certainly cells in the laboratory, hasn't changed for probably over 100 years. It's almost the same thing. What we've brought forward now is completely new ways of doing this, which make them more effective and more reproducible.
Yep, cool. And I did see a bottle of your product. There's a little pink bottle. And so is it one bottle per experiment, or does that bottle go a long way? So
a bottle that's a 25 ml bottle. That's a 4x concentrate. That will give you, depending which format you use, anything up to 1,000 experiments.
Wow, that's amazing. Yeah, that's huge. And are you manufacturing that in Australia?
Yes, it's being fully manufactured in Australia and sold here. Yes.
Excellent. I was really happy to see and glad to see that you have really thought about the IP protection. So that's really good. Heaps of patents there. Anything else that you want to add to that?
So, it's where most of our money's gone over the last 3 years. We've spent a huge amount of money protecting this, and not just getting patents, but making sure that these are bulletproof. We've had some multinationals already try and steal our technology. I won't mention their names, but very, very large American company and a very, very large Japanese company have actually tried putting in competing patents and have failed. So we've got a very clear IP landscape at the moment, and we're very, very proud of it. Actually, we've been very lucky to get the sort of level of protection that we have with these patents.
Yep, exactly. And just last question is, if you were to give one bit of advice to a person or a company who's thinking of getting into like biomed or biotech, what would you say to them? What sort of advice?
Well, probably it's not going to be the right advice. It's probably not going to be very politically. So I'll be honest with you, stay away from therapeutics. Do not invest. I would be very skeptical, or very. I'd be I certainly wouldn't invest in therapeutics or anything that requires any FDA approval or anything. There's a lot of money that's going to get spent. Most therapeutic compounds, there's a huge attrition rate. So to be honest with you, you'd be better off putting your money on a horse than putting it on therapeutic. But that's just my opinion.
Anthony, can I ask you a question about one of your slides?
Yeah, sure.
Maybe I'll get you to share your screen again, if that's okay, and flick back to the slide which had the graphs on it. There was some very interesting graphs on the slide. There was like 3D graphs. You know, that one about the reproducibility of testing, I noticed.
Oh, sure, yeah.
One of that graph there. Maybe if you can just spend a minute on just explaining that graph, it would look very interesting.
Can you see that?
Yes, it is just starting. Yep, that's the one.
So variability with in the sort of cellular environment is really sort of centers around three factors. One is temperature. The other is osmotic or water concentration. So the amount of how concentrated your media, the growth media, or it's almost like a blood-like liquid that you keep the cells in, and also pH. So what we show here is firstly that our technologies stop the cells from cooling down. So cells are optimal at around about 37. But if you take a microplate or one of these systems out of the controlled temperature environment to perform some experimental procedures, they get cold very quickly, and this can cause huge problems with the cells. So this one just shows us here that the cells retain, our microplates retain heat 3 times longer than a standard microplate system. The second one is an extremely compelling experiment. So evaporation is a huge problem. If you get evaporation in your microplate system or with your cells, what happens is that when the water leaves the solution becomes more concentrated, and that changes the concentrations of salts, if you're using drugs, all sorts of things. And this can cause huge problems, physiological problems for the cells. So what we show here is that the top trace is our plate, which was put into a drying oven. So we filled a plate with liquid, we put it into a drying oven for 48 hours at 50 degrees. And this shows you the plates here without our technology in it, an identical plate. And as you can see, what we have here, we've sampled across the plate. What we see here is that the corners have lost almost 80% of their liquid, and even the middle has lost up to 20%. That's still far too much liquid to lose, whereas our system doesn't lose, virtually loses nothing, maybe just a few percent in the corners. So our technology here prevents environmental changes, environmental fluctuations, which cause problems with cells. Now, leading on here, these traces here show this is actually cell number. So what we've done, we've seeded plates, we've left them in the incubator for a week under normal conditions. And this plate here, or this experiment here, was done on a normal plate. And we've got a coefficient of variation of 12.7%. And taken with our plates system, our microplate system, we've halved that coefficient of variation to 6.12. Depending on the cell type and the experiment you're doing, these coefficient of variations can be very much higher. Down here showed this is a very compelling experiment. This was done with a 12-well plate, which is the system we're currently selling. This system here is a 384 well, which is the industry a 384 well plate, and this is the industry standard for the pharmaceutical companies. And here we can take our coefficient of variation. You can see the huge variation between the outer and the wells here. And we can take that from 31 down to 13%, which is unprecedented. People have never seen this sort of improvement before under these sorts of experiments. So it takes away all those problems associated with variation within a plate. So if you imagine, this is a 384 well plate. So there's 384 little Petri dishes in that plate, in that SBS format plate. Each one of those will contain a candidate or a test drug. If you put your cells or you test drug in one of these outer wells, it's going to give you a completely different readout from if it had been put into one of the middle wells. So we basically flattened that out to make sure that the variation is very much smaller. And I think we can even improve on this. These experiments were done some time ago. And now with improvements in liquid handling and cell dispensing, we can actually flatten this even more. I hope that explains the experiment.